Natalie, what do we age? We have oxidative stress, epigenetic dysregulation, DNA damage, mitochondrial dysfunction, but today we're only talking about one telomere shortening. Here we see a double-stranded canonical representation of a DNA. These strands are complementary to each other as a template for the other half. We can see a leading strand and a lagging strand, both organized in a palindromic fashion. This simply means that the leading strand has its nucleotides orientated in a 3' to 5' direction, and the lagging strand has its nucleotides orientated in a 5' to 3' direction. A RNA primer with a free 3' hydroxyl group is placed to initiate replication. An enzyme called DNA polymerase can then add on matching nucleotides for the leading strand. However, because DNA polymerase can only work in a 3' to 5' direction, several primers have to be added for the lagging strand to be built in a backstitching mechanism, so polymerase can work in 500 to 1,000 base pair chunks, known as Okazaki fragments. When RNA primers are later removed by a degradation enzyme, RNase H, the gaps between the fragments are filled in once again by polymerase, then joined by ligase by recognizing the hydroxyl groups in the individual fragments. But since there are no hydroxyl groups here and here, both the lagging and the leading strand will be shortened after replication. Naturally, the body will not allow for important coding material to be placed at ends because after incomplete replication, these may be lost. So instead, telomeres, which are shoe caps of non-coding DNA, shorten in place of these coding genes. These introns are crucial in preserving chromosome integrity, and without them, our cells are unviable. The number of times that a cell can divide before cell death is known as the Hayflick limit. This number differs in the species of organisms which justify their lifespan. For example, mice who have a Hayflick limit of only 14 to 28 can only live up to 2 to 3 years, but galloping giant tortoises who have a Hayflick limit of 125 can live more than 100 years. And since telomere shortening directly causes aging and senescence, it is important to understand it thoroughly, and you should research it more.